Hi, I'm Maider, and today I will be discussing the KRAS gene, its significant role in cancer development, and the ongoing challenges and breakthroughs in targeting it for therapy. The KRAS gene, a member of the RAS gene family, is crucial for regulating cell division, growth, and apoptosis. However, mutations in KRAS are among the most common oncogenic drivers in human cancers, particularly in lung, colorectal, and pancreatic cancers. These mutations often lead to aggressive cancer forms with poor prognosis, making KRAS a challenging but critical target for cancer therapy. KRAS functions as a molecular switch, cycling between an active GTP-bound state and an inactive GDP-bound state. Its role in the MAPK-ERK signaling pathway is to transmit signals from the cell surface receptors to the nucleus, regulating gene expression, cell proliferation, and survival. In normal conditions, KRAS ensures that cells divide only when necessary. However, mutations in KRAS lock the protein in its active state, leading to uncontrolled cell division, a hallmark of cancer. These mutations are particularly common at codons 12, 13, and 61, with its mutation driving oncogenesis in slightly different ways. To understand the impact of KRAS mutation, let's look at some statistics. In non-small cell lung cancer, also known as NSCLC, about 30% of cases involve KRAS mutations, with G12C, G12D, and G12V being the most prevalent. In colorectal cancers, KRAS mutations are present in approximately 40% of cases, and in pancreatic cancers the rate exceeds 90%. These mutations lead to continuous activation of downstream signaling pathways, particularly MAPK-ERK and PI3K-AKT, which promote cell proliferation, resistance to apoptosis, and enhance migratory and invasive capabilities. As a result, cells with KRAS mutations no longer rely on external growth signals, allowing them to grow uncontrollably and form tumors. Here's a diagram illustrating how mutant KRAS continuously activates the MAPK pathway, bypassing normal regulatory mechanisms. The consequences of this persistent activation are severe. It not only drives tumor growth, but also complicates efforts to target KRAS for therapy because the pathway remains active regardless of external signals. Targeting KRAS has been notoriously difficult due to its high affinity for GTP and lack of suitable binding pockets for small molecules. Historically, this made KRAS an undruggable target in cancer therapy. However, recent breakthroughs have changed the landscape. The discovery of covalent inhibitors that specifically target the KRAS G12C mutation marks a significant advance. Sotorasib and adagrasib are two such inhibitors that have shown promising results in clinical trials. These drugs work by irreversibly binding to the mutant cysteine residue, effectively inhibiting KRAS's ability to signal downstream. For instance, in the CodeBreak 100 trial, sotorasib achieved a response rate of 37% in heavily pre-treated non-small cell lung cancer patients with a median progression-free survival of 6.8 months. This is a remarkable achievement, given the aggressive nature of these cancers. Continuing talking about sotorasib in patients with non-small cell lung cancer harboring the KRAS G12C mutation, sotorasib has shown a significant reduction in tumor size and improved progression-free survival. These results not only highlight the potential of KRAS-targeted therapies, but also underscore the need for continued research to expand their efficacy across different KRAS mutations. Despite these successes, challenges remain. Tumors often develop resistance to therapy, finding alternative pathways to continue growing. Additionally, expanding the efficacy of treatments to other KRAS mutations such as G12D and G13D is still an ongoing area of research. As we look to the future, one of the biggest challenges is overcoming resistance to therapy. Tumors are highly adaptive, and the development of resistance can limit the long-term effectiveness of KRAS inhibitors. Researchers are exploring combination therapies that pair KRAS inhibitors with drugs targeting other pathways. Moreover, emerging technologies like CRISPR-Cas9 for gene editing and RNA interference hold promise for directly targeting KRAS or its downstream effects. These approaches could revolutionize how we treat cancers driven by KRAS mutations, offering new hope for patients.